Well, hello. I think we're loading. Hello, AP Bio students. Mrs. Shelton here. Today we are going to be talking about unit number 5. So this is unit number 5, AP Bio Heredity. This is your textbook chapters 10, 11, and 12. And your test prep review book, this is topic 4, is what we should be looking at here. Now, in Unit 5 in Heredity, the way College Board actually breaks this particular unit down is they are actually talking here about meiosis and making sure you understand basic Mendelian genetics and also non-Mendelian genetics, and also then understanding our chromosomal basis of inheritance. And then we'll carry this on to our last unit that we'll cover for this year, which is Unit 6, which then we'll talk about gene expression and regulation. But we'll just talk about the inheritance patterns today. Now, in preparing for the AP exam, some things that College Board points out is that we need to be making predictions about chromosomal exchange, especially talking about crossing over. When we talk about genetics, obviously being able to calculate our expected genotype and phenotype outcomes and ratios, that's important. But they're also emphasizing here that you should be able to calculate chi-square values when we're actually performing a cross and being able to compare that observed and that expected. Also being able to understand how to identify the null hypothesis. And so when do we accept or reject that null hypothesis? So we'll make sure that we review that again as well. Now, some of the big idea questions, because like I've told you that all of these units, even though they're not covering unit 7 and 8 on this year's test, we still have to make sure that big idea number 1 is still incorporated and we understand how it's incorporated throughout all of our units. So obviously, when we're talking about genetic inheritance, this is a huge impact on evolution and natural selection. So how do we move information from one generation to the next? Also, when we talk about information storage and transmission, how do we pass that information, that DNA on? And then this question is, how would Mendel's laws have been affected if he had chosen something other than pea plants to study? Because what you have to remember is that Gregor Mendel actually got lucky in the fact that with pea plants, all of the 7 characteristics that he was studying, they were all either so far apart on chromosomes that they weren't linked, and they all behaved independently. Also, there was nothing other than just pure dominance and recessiveness in those characteristics he was studying. And then big idea number 4, diversity. And we'll talk about how we generate diversity, but then also carrying this on to Unit 8, because in Unit 8, we talked about the importance of genetic diversity in an ecosystem. And so here we need to make sure we understand how we obtain that genetic diversity in the first place. So those are your big ideas. Now, overview of what we're covering in this unit, meiosis and genetic diversity. Here's our genetic sections 3 and 4. And then we're also going to be addressing how does environment also affect phenotype, and then this chromosomal basis of inheritance too. So those are some of the major concepts to be addressing. Now in 5.1, 5.1 is your conversation about meiosis and making sure we're understanding with meiosis that it undergoes 2 rounds. Meiosis, that we start with a daughter cell, we're always reducing our chromosome number in half. So we are starting with a diploid nuclei and we are reducing it to 4 haploid nuclei is what is happening in this. We also have to be able to compare and contrast mitosis and meiosis. So those are our first concepts here. So when we look at meiosis, some events to make sure that we are recognizing in meiosis is, first of all, in this first round of meiosis, the goal of the first round of meiosis is to separate homologous pairs. So I'm separating my homologous chromosomes in my first round. So that's a huge difference that I have between mitosis and meiosis, is the fact that I have homologous pairs that line up on the metaphase plate. My sister chromatids are not separating at this point. Now, also what happens in my first round of meiosis is crossing over occurs. This is a huge, huge impact on genetic diversity. And then I also have my independent assortment based on how these homologous pairs line up on the metaphase plate. I'm going to have independent assortment occurring as well. So ultimately, what is produced are 4 haploid cells that are all genetically different from each other. So just in meiosis alone, so it's in this first round of meiosis, here I'm going to have my crossing over event, here I'm going to have my independent assortment. My first round of meiosis, I'm separating my homologous chromosomes. My second round of meiosis, though, now I'm going to have my chromosomes. They look very similar to what metaphase looked like in mitosis. They're just lining up single file, and it is now in anaphase 2 that sister chromatids now separate, creating now my 4 haploid nuclei. So that's the goal of meiosis, is to reduce chromosome number. So remember in mitosis was creating identical nuclei. So I reviewed some of the differences. Meiosis, we're creating haploid cells. We're going from a diploid state to a haploid state. We're doing it in 2 consecutive rounds. Remember that also in prophase of meiosis, I have crossing over that occurs. Also remember that in metaphase 1, I'm going to have my separation of homologous chromosomes happening as well. And then in mitosis, I'm going to start with a diploid nucleus. I'm going to end with a diploid nucleus. In meiosis, I'm going to start with a diploid nucleus, but I'm going to end with 4 haploid nuclei. Here I go from 1 to 2. Here I go from 1 to 4 cells is what is happening. Do remember, however, the DNA content, though, is doubled initially because that S phase of the cell cycle has already occurred. So remember here, I'm seeing 4 chromosomes, but I am seeing my chromatid still attached to its sister chromatid at the centromere. So remember that. So I have 4 chromosomes here. I have 4 chromosomes here, and they're showing you the parent cell before chromosome replication. So remember how to count your chromosomes in biology. So mitosis and meiosis, make sure you are knowing the difference between them. Now, as far as meiosis and how it establishes genetic diversity, I've beat this into your head, so this is a super easy thing to make sure that we are going over. Anytime meiosis occurs, we are obtaining genetic diversity in the new cells that are produced because of crossing over and independent assortment. So with that, we even talked about with independent assortment alone, just the number of combinations of gametes a human could produce just with independent assortment alone. So then add crossing over in there, and we just get tremendous amount of diversity and a genetic recombinations that are occurring. Now, also then you add in random fertilization. And now with that, all of those millions of combinations, so I had millions of possibilities for an egg being produced, and then millions of possibilities with a sperm, and then it's random which of these gametes fused together can now produce a zygote. And so this is where our genetic diversity comes from. So our genetic diversity through meiosis and sexual reproduction is going to be through, in meiosis, we're going to have our crossing over, our independent assortment, and then in sexual reproduction, I'm going to have the fusion of my gametes, my 2 haploid gametes, to reestablish a diploid state, and that's going to give me genetic diversity. So 5.3, Mendelian genetics. So this particular part up here on this learning objective, they're addressing for you here, making sure that we are talking about genes, we're talking about genetic information, DNA and RNA. We will talk more about this in Unit 6. So at this point, we'll talk about our transcription, translation, and those shared processes there. In this particular review, I really just want to make sure that we are addressing basic Mendelian laws of law of segregation, law of independent assortment, making sure that we are calculating probabilities correctly, understanding our law of probability, making sure we understand that these probabilities of creating these zygotes, what a monohybrid cross looks like, a dihybrid cross, sex-linked traits, what linked genes are, how do we calculate this data for genotype and phenotype as well. So that's really the important part that we focus on here, and practicing some different genetics problems too. Now, some terms to make sure that we are clear about is when we talk about a monohybrid cross. Remember, a monohybrid cross is going to be this cross between two hybrids. So here I have a heterozygote parent crossed with another heterozygote parent. And what you absolutely positively must make sure that you memorize is my expected phenotype ratio is a 3-to-1 expected phenotype ratio. My expected genotype ratio then would be the 1-to-2-to-1. So make sure that you are remembering your basic genotype and phenotype ratio expectations. In a dihybrid cross, now I'm tracking the inheritance of two different genetic traits at the same time. But remember here, when now I have this particular yellow smooth seed that's heterozygote for color and for shape, I still have law of independent assortment. So I could be passing on a dominant allele for shape and a dominant allele for seed color, but I could also have the dominant allele for shape with the recessive color as well. So it's making sure we understand that from this one parent, I have 4 possible gamete combinations that could be attributed here. Now, when I have a dihybrid cross, College Board is not going to expect you guys to do a 16 square Punnett square, but they are going to expect that you understand how to use rules of probability. So if I have a probability here and we'll talk more about probability in a little bit, but making sure that you could multiply your rules of probability. Now, basic thing to make sure that you are remembering from a dihybrid cross, my expected phenotype ratio in a dihybrid cross is 9-to-3 to 3-to-1. So always make sure that you are remembering a true dihybrid cross expected phenotype ratio is 9-to-3 to 3-to-1, 16 different from those 16 different possibilities of what we are determining in those phenotypes, 9-to-3 to 3-to-1. Now, we should make sure that we are understanding pedigrees and how do we determine from a pedigree what type of inheritance pattern it is. Is it autosomal dominant? So is it carried just on an autosome, a non-sex determining gene, or is it sex-linked? And how we determine that is it affecting males and females differently? If it is sex-linked, it is going to be affecting males and females differently. If it is autosomal, it's going to be just males and females are equally affected. Now, if it is a dominant, autosomal dominant characteristic, then what we would expect to see is if it's autosomal dominant, always look to see where you have two parents that are the same and a child that is different. So in this case, if I have two parents that carry that dominant trait, they could have a child that does not. If it's autosomal recessive, you have parents that aren't showing that trait, but they have a child that does. So that's how we would look at autosomal recessive. Now, X-linked dominant. The only ones that we're going to be looking at is our X-linked recessive. And an X-linked recessive, what you are going to be seeing is one, you're going to see the males being affected more so than the females. So that's the example that you would be looking at and being able to determine that pattern of inheritance from a pedigree. Now, in our probabilities, the types of probabilities that we did so often is we use the rule of multiplication. And when we use the rule of multiplication, when we do that is we're looking at 2 or more events happening at the exact same time. So I like this explanation here is if they're saying, what's my probability of this and this happening at the same time, then you multiply your individual probabilities together. Now, if we're talking about a rule of addition, here you're looking at an outcome that has different ways of getting there. So a key word to look for if I'm going to use my rule of addition is the or part of it. So here they use the example of my probabilities of having offspring with a dominant phenotype. Well, here we would add up my 3 different ways. I have a 1/4 probability of getting homozygous dominant. I have a 1/4 chance of getting a maternal dominant allele and then a paternal recessive, and then I could also get heterozygote the other way. So I add all those up in order to give me my probabilities. So that's when I use the rule of addition if they tend to be different events that are happening, keyword or, if there's two ways to be heterozygous. Now, also in this section here, and I'll give you practice problems that we address. Again, with genetics, genetic outcomes are a great opportunity to now practice your statistical analysis of performing chi-square testing. This is directly cut from the data sheet that College Board will be providing for you. Remember, here is your chi-square analysis equation. Remember, it's a summation problem where you are now taking the difference between your observed values, so what you really got in the lab, minus your expected. Your expecteds would be calculated from your probabilities, and you square that value divided by the expected. Now, it's a summation, so there's multiple outcomes that we would expect. So maybe we're looking at green and yellow as my possible outcomes. Well, in that case, remember that the number you calculate for chi-square is just a number. You have to then be able to compare it to one of your critical values. Your critical values are these numbers that are listed down here in this chi-square table. They're not telling you that these are the critical values. You need to know that my possible critical values are 3.84, 5.99, 7.82, and how you know which critical value to use is based on your degrees of freedom. In the example I just said, if I was looking at yellow versus green offspring, well, I have two probable outcomes. So I'm going to have two different outcomes that I sum together. My degrees of freedom are probable outcomes minus 1. So I would use 3.84 to compare to my chi-square value that I calculated. Remember that I always use a p-value of 0.05. I never use this line down here. That's a distractor line, so you can just cross it out and never just pretend it doesn't even exist. But chi-square analysis, we'll make sure we do some practice problems again on those, so you feel comfortable if they ask you to do that type of data analysis. Now, in non-Mendelian genetics, what we're talking about here with non-Mendelian genetics is understanding that linked genes. So anytime I don't get those expected outcomes of a 3-to-1 phenotype ratio in a monohybrid cross, or I'm not getting my dihybrid cross of 9-to-3 to 3-to-1, that's when I have to look at what other modes of inheritance could be impacting that outcome. So with that, some things to make sure that we are addressing, and even, like on here, we're talking about multi multiple gene traits. We're looking at polygenic inheritance. We're looking at linked genes. We're looking at codominance. Epistasis would be another example. So all of these are non-Mendelian genetics that we should be looking at. And then also remembering too that chloroplasts and mitochondria have their own DNA that also give different phenotypic effects as well. And that's all inherited from the mother. So recognize that it's not just purely dominant and recessive inheritance on autosomes that we need to be focusing on. So if we're taking a look at some of these non-Mendelian genetics, sex-linked traits would be one that we need to make sure that we can calculate different sex-linked inheritance, because here again, we have to, if it's sex-linked, that means the recessive trait is carried on the X chromosome, and so it's going to be affecting males differently. The Y chromosome does not carry the same information as the X chromosome. So what males get on their X chromosome from their mother is what always shows up. And so you need to make sure that you are showing that, like for hemophilia or for color blindness, you are showing that recessive trait carried on the X chromosome. Now, in this particular case, this happens to be a cross between a dad who has hemophilia and a mom who's a carrier. So with this, we are seeing that in this case, I have one daughter who has hemophilia, and I have one son that has hemophilia, and then I have a second daughter who's a carrier. Now, in this example up here, this might be something that you guys are forgetting, but this would be our blood type. Remember, blood type is an example of a multiple allele trait because there's 3 different genes in our gene pool, but it's also an example of codominance. So where the A and the B are codominant to each other, but the O, which gets written as a lowercase letter, the lowercase letter being the fact that it's recessive to both A and B. So this would be a mom who was type A blood. This would be a dad who's type AB blood. So what they would have told you is that this mom is type A blood, but her mom was type O. Because if her mom was type O, her mom was lowercase I, so you know that she had to have inherited one of those lowercase i's. Now, in this case, they're showing you the Punnett square here and showing you all the probable outcomes of a dad who was type AB blood and a mom who was type A blood, but who had a parent who was type O blood. And these are their possible offspring. Now, another example that you should be familiar with is incomplete dominance versus codominance. Now, remember with incomplete dominance and codominance, you would actually solve these problems. So we would write our codominance just like this. We would write a capital letter with a superscript. The difference between incomplete dominance, though, and codominance is just what it looks like in the phenotype. In codominance, I can see the different phenotypes showing. So in blood type, it's codominance because I see the A and I see the B antigens still on that red blood cell. In incomplete dominance, it's a blending that ends up happening. So in the phenotype, I don't see the red, nor do I see the white. I see now the blending where it all looks to be now this third phenotype. And that's where we'd also know if it's incomplete dominance or codominance, if my genotype ratio and now my phenotype ratio are both the same. If my genotype ratio is 1 to 2 to 1, and now my phenotype ratio is also 1 to 2 to 1, then it's incomplete dominance or codominance. And you would solve those genetics problems the same. It's just the difference in what the phenotype looks like is the only difference. Polygenic inheritance would be another example of something that's non-Mendelian. So in polygenic inheritance, so this is trying to show you with polygenic inheritance, if I have 3 genes that all impact now pigmentation. Well, the different combinations of how those 3 genes interact now actually give us this bell curve result in all of the offspring. So with polygenic inheritance, there's multiple genes that contribute to one phenotype. And so that is where I'm now getting this bell curve type of result that is occurring. Remember too, linked genes. If genes are linked, they are found close together on the same chromosome. They often get inherited together. But how far away they are from each other on the chromosome increases their chances of recombination due to crossing over. But also remember too, if genes are located on opposite ends of the chromosome or far away, then they do behave independently and they have a frequency of recombination of about 50%, just like they were on different chromosomes. But with linked genes, if I start seeing 2 particular traits always being inherited together, even though I'm looking at a dihybrid cross, well, that would be my evidence of linkage. Now, it's through linkage that we can also determine a gene map, and it's your frequency of recombination. So here's my recombination frequencies, what are given to us with these 3 genes. So the frequency of times that these 2 genes recombine to give a dominant and then a recessive when we're doing dihybrid crosses. And so through those, this tells us now locations on that particular chromosome. The higher the frequency of recombination, the farther away those genes are. The lower the frequency of recombination, the closer together those 2 genes are on the same chromosome. So remember how crossing over can also give us clues as to gene location and determining gene maps. And then lastly, in this particular section, it's remembering that mitochondrial DNA, all of your mitochondrial DNA came from your mother. So thank you, Mom. And chloroplasts also carry their own DNA. And so these do have an influence on now some different phenotypes, is what mitochondrial DNA you inherited, or if you're a plant, what chloroplast DNA did you inherit from the ovum of the female. Some illustrative examples. So when we talk about sex-linked traits, be familiar with the fact that it's not simply just like humans where females are XX and males are XY. Do just recall that there are other types of sex determination that can occur. If College Board is asking you to solve a problem with a different type of chromosomal inheritance, so like in chickens, where now it's the female that is hemizygous and the male is homozygous for sex determination. Or if we're talking about bees, where bees actually have males that actually have, so now we have a haplodiploid system here where males actually have half the chromosome information. They only are these haploid state versus now a diploid state, or they can also be XY. So College Board will always describe that for you if they are giving you another type of um, sex chromosome situation other than the standard human or most mammals, which are the XX and the XY for females and male system. Now, in environmental effects on phenotype, this was super, super easy. We addressed this pretty quickly, but it's recognizing that phenotype is not just determined through genetic inheritance alone. Environment plays a huge role on that influence. So for this, there's just lots of illustrative examples. So in here, these twins, amazing, amazing brothers, but they are identical twins, but they don't look the same. One actually spent over a year in the International Space Station. Different environmental impacts have led to different weights that they carry, different physical shapes that they have as well. We also talked about just pH. So these hydrangeas. So all these hydrangeas have the exact same genetic makeup, but the soil pH actually affects their pigmentation in their flowers. Seasonal fur in Arctic animals. So here, why would the Arctic animals want to be white in December? Because that's the time there's snow on the ground. And with snow on the ground, now there's more camouflage and they can blend in. They can also absorb more light as well in order for maintaining body temperature. But here in the fall, when there's no snow on the ground, well, now they're darker in color. So, but it's environment that's actually shaping that pigmentation that's occurring. Sex determination in reptiles. Well, different temperatures that the eggs are developing under actually changes the gender. So here's a graph that's actually showing that. And there has been an AP question in the past where it gives you a graph of the different reptilian eggs in different temperatures. And with that showing different genders that are being determined. So here's the sex ratios that they're actually showing based on temperature change. And here's just a quick little easy one to understand in this clutch that's laid down here in the colder temperatures. These are going to be female. The ones in the middle that are the medium temperature, those are going to develop to be male. The ones up top that are more in the hotter range. So that's very similar to this type of curve that you're seeing there. We all know that UV light can trigger melanocytes to produce more melanin. So we can actually have more skin pigmentation when the UV light is stronger in order to protect your cells from UV damage. I'm leaving out this one because as long as you're understanding these illustrative examples, trying to explain fungal reproduction and pheromone influencing on it, I'm going to skip it. And then 5.6 is addressing chromosomal inheritance. So with this, again, we're going back to segregation, independent assortment, making sure we understand fertilization, but then also making sure we can relate genetic disorders to this chromosomal inheritance in sexual reproduction. So when we do talk about genetic diversity, remember again, our sources of genetic diversity, we do have mutations, so we'll talk more about mutations in our Unit 6, but mutations do provide different variations of genes. But remember that through meiosis and random fertilization, It's the crossing over, independent assortment, my random fertilization. This is what is going to give me the majority of the genetic diversity that we see in populations. If we look at this diagram, you should all recognize that this is a karyotype, and so this is a human karyotype that is showing our 22 pairs of autosomes, our 1 pair of sex chromosomes, and these chromosomes here, chromosome pair number 1, these are homologous. Remember, homologous chromosomes are not identical, but they carry the exact same types of genes at the same gene loci. So if this was the gene for hair color, then hair color would also be over here. But the genetic code for hair color here might be for brown. The genetic code for hair color over here might be blonde. Now, do remember that at this point, when I'm taking a karyotype, my chromosomes have already replicated, so the S phase has already happened, and so I have a chromatid attached to its sister chromatid. They're much more visible this way. But this chromosome is homologous to this one. They're not identical. They're just similar in genetic information they carry. They're similar in length, and then you also see in the banding patterns, the striping patterns, because they're carrying similar genetic code. Now, I have 22 pairs of autosomes, 1 pair of sex chromosomes. This is a female, and this is a normal female with a normal number of chromosomes with the 2 X chromosomes. Remember that Y chromosome? Y chromosome carries that SRY gene that would influence the tissue development of testes that is actually of testicles that's carried on the X chromosome. But there's no Y chromosome, so this is a female. Some illustrative examples to just make sure you're understanding inheritance of genes. Sickle cell anemia, we've talked about a lot. This is a classic example that College Board likes to address. Remember that the sickle cell trait, this is also an example of heterozygote advantage. These two parents being heterozygous, this is a autosomal recessive disorder, but it demonstrates heterozygote advantage. These two parents actually are resistant to malaria because they're carriers of the sickle cell trait. But in this particular case, now I've got, this is my monohybrid cross, and in a monohybrid cross, it's a 3-to-1 phenotype ratio, where in a 3-to-1 phenotype ratio or a 1 to 2-to-1 genotype ratio, I have a 1/4 probability that these parents will have a child who has sickle cell anemia. And in sickle cell anemia, now red blood cells, just due to pH changes and pressure changes, now these red blood cells, because of the difference in the hemoglobin, can change into this sickle shape and can clot. So remember that for sickle cell. Now, Tay-Sachs disease. In Tay-Sachs disease, Tay-Sachs is also autosomal recessive. And with autosomal recessive, I still have that 1 to 2-to-1. If both parents have to be heterozygous, they have to be carriers. And so I have a 1/4 probability that they would have a child who has Tay-Sachs disease. Now, Tay-Sachs disease, unfortunately, what happens is a faulty enzyme. And so this is a lysosomal storage disorder. And so what ends up happening is the baby actually is born normal, but over time the neurons start breaking down and stop functioning, and usually death is going to occur somewhere by age 4 is kind of the average life expectancy of a child who's born with Tay-Sachs disease. Now, Huntington's disease, however, is your example of an autosomal dominant disorder. We do not have many dominant lethal disorders in organisms. And the reason why is if it's lethal and dominant, then you're not passing it on. But remember with Huntington's disease, this happens to be a neurodegenerative disease. But with this, remember that the age of onset usually doesn't happen until after reproductive age. So now with genetic testing, however, people are recognizing that they have Huntington's disease and are making a choice not to reproduce, or they're screening their offspring is what they're doing with Huntington's disease. But that would be your example of one case of an autosomal dominant lethal disorder. Now, X-linked color blindness would be something that we've already talked about, but it's making sure that you could complete the Punnett squares if you were asked, making sure that you are showing on your Punnett square and calculating your probabilities, you're showing this condition. It's recessive, so it's X-linked recessive. carried on the X chromosome, so you have to show how it would be affecting males and females differently, is what you would have to be showing. So in this case, if I have mom who's a carrier, dad who's normal, in this case, this is where I can have daughter who's now a carrier. So half of my daughters are carriers, but half of my sons are colorblind, is what's going to be happening here. And then lastly, we should be familiar with non-disjunction disorders. And in a non-disjunction disorder during meiosis, and this could happen during meiosis 1 or meiosis 2, it's a failure of chromosomes to separate. So if I have a failure of chromosomes to separate during meiosis, then in non-disjunction disorders, I can have extra or missing chromosomes. So in this particular case, I have an extra 21st chromosome. And so in this is what Down syndrome is. Down syndrome is an extra 21st chromosome. This is what it's called trisomy 21. But those are my non-disjunction disorders. We also talked about, I'm not sure what's going on here with this X chromosome and this Y, because they just drew those funky. Bad diagrams I chose. But remember, we also talked about Klinefelter's. We also talked about Turner syndrome and some other non-disjunction disorders that affect sex chromosomes. But understand some of those chromosomal bases of inheritance too. So that is your Unit 5 in a nutshell. Practicing genetics problems, going back to your test prep review would be highly recommended, is what I would suggest that you guys do. And then with that, we have just one more unit to cover, Unit 6, and we'll cover that, and then we'll just practice, practice, practice until we take the AP test. So anyway, I will talk to you guys soon. Miss you. Wish we were in class together, but hope this helped in your studying and helping you with your focus. So talk to you guys later.